Okay, let's roll. So we last left off right in the middle of translation. So just to refresh your memory, we just got done talking about initiation events. And so now we're in elongation. And we kind of sketched out elongation a little bit, right? There's three primary steps to elongation, which is codon recognition. And then, of course, that breaks into peptide transfer and then into translocation. So if we take a look at codon recognition, basically, we're going to start down here. And so in codon recognition, you notice you've got an empty A site at this particular point. So what's going to happen is your elongation factor is going to be bringing in the correct tRNA and pairing it up with that codon, right? So that's the codon recognition piece. So that's where the tRNA and the mRNA meet each other. So once the elongation factor has finished its delivery, it's locked into place. And at that point, you now have a complete A site with your new tRNA in it. And then, of course, you lose your elongation factor. So it's done its job, right? Shuttling that tRNA to its correct site. So that's basically where your tRNA will bind to your codon in your A site. Then once you're in your A site and you have your amino acid, now what's going to happen is you're essentially going to transfer the peptide chain to the new amino acid. That's where you get your peptide transfer. So notice what happens is you have your growing chain here, which is 2 at this point. And what's going to happen is you're going to take that chain and you're basically going to add it to the new amino acid. So now at the end of peptide transfer, you now have a peptide of 3 amino acids, right? Because you added the first 2 to the new one. So that gives you 3. But now, if you take a look at it at the end of peptide transfer, your spacing is all wrong, right? Because the tRNA that's holding the polypeptide is now in the wrong site, right? Because that site, the peptide site, is supposed to be in the P site. Right now in the P site is the empty tRNA. Well, that one needs to be in the E site. That's what the E site is for, right? And the A site should be empty. So everybody seems like they're displaced by one site right now. And so in order to solve this problem, we go to the last step of elongation, which is translocation. So it's a simple procedure. All you do basically is take your mRNA and you essentially pull it that direction. What that's going to do, it's going to shift the empty tRNA into the E site where it belongs. It's going to shift the polypeptide tRNA into the P site where it belongs. And then it's going to open up your next codon in line to be read. Now, after translocation, everybody is in their proper place. The A site is empty, ready for the new delivery of the new tRNA in line. The P site is full of the tRNA with the polypeptide chain in it. And in the E site, you have your spent tRNA, which will get ejected. And then this will go and get recharged by going back to that amino acyl tRNA synthetase to get another amino acid attached to it. And then at this point, you start it all over again. You do codon recognition for the next codon in line. And so you continue this cycle over and over and over again until you hit your termination signal, which is easy in translation, much more so than other things. So it's kind of an up close look at peptide transfer. So if you take a look at peptide transfer, basically what happens is when the new amino acid comes in, the amino group of the new amino acid actually attacks the carboxy carbon. When it does that, it forces a bond between these two. That becomes your peptide bond. And then that sufficiently links. Then this will basically move to the backside of the growing chain. And then that sufficiently links your 2 amino acids together. But they're going to be linked together on the new tRNA. So notice what happens. When you're growing your chain, it's a little bit counterintuitive, right? Because the way we form lines is by the new person shows up and they go immediately to the back of the line, right? If they go to the front of the line, they're probably going to get themselves beat up. Okay. So you don't go to the front of the line, you actually go to the back of the line. In this case, it's a little bit different. In this case, it's like you show up to this big line, and what happens is the line gets behind you. So everybody who shows up new is immediately at the front of the line until the next person shows up. Then they get behind the new person. Okay. That's how transfer is working. And when you look at the organization of the transfer, you notice as you get your growing chain, on this end, you're going to have your amino group. And connected to the tRNA is going to be your carboxyl group. So as the chain grows, you basically continue this. So when you take a look at the growing chain, you can see that you have your amino group here, which you call the N-terminus. And you have a carboxy group down here, which you call the C-terminus. And in between, you have your series of amino acids from 1 to however many you've got. In this case, 7. And that's true for every protein, right? So whenever you look at that primary structure, right, just the linear chain of amino acids, there's always an N-terminus that starts the first amino acid and the C-terminus, which is the last amino acid. Okay, so one of the things that we noticed, however, about this process is there's a little bit of flexibility in there. And so what we saw was what we call wobble pairing. So if you take a look at the anticodon-codon relationship, right? So the 3 pairs, let's imagine we had an anticodon that looked something like this. Normally, you would expect that to be paired with an A, right? And a U in the first 2. These 2 here seem to have stringent pairing rules. That is to say that you have to be an A and a U, right? However, this one is not so stringent. So there's some flexibility in this one. So for instance, this could be, normally it's a G, right? But this one could be, instead of a CG, it could be also a CA or a CT. CU rather. So there's a little bit of flexibility in there. There's some slush in that third base. We call that wobble pairing. It kind of wobbles around a little bit. What it does is basically it allows you to be able to preserve the assignment of your amino acids without necessarily having to be so stringent in that third one. All right, because think about it. The stringency of the first and the second is sufficient to impose the selection that you need. You've got what you got, right? You've got what you need with the first 2. So you can slop it a little bit with that third one. The benefit of this is it allows you to be able to economize. So rather than making a tRNA for every single selection out there, it allows you to use fewer tRNAs so that you can cover all your codons with not as many tRNAs, because otherwise you're going to be spending a lot of money making tRNAs for every single codon pair out there, right? So this is kind of like an economy thing, right? If I can get the job done with half as many tRNAs, then that's good economy, right? And that's basically what this is. It gives you what you need, the specificity that you need for a little bit of a cheaper price. Okay, so it's a value pick. This is a value pick. That's what this is, wobble pairing. Okay, termination is easy, right? Because basically you hit a stop codon. Either a UGA or a UAG or something of that nature, right? And so what happens when you hit a stop codon? Well, when you hit a stop codon, what's going to happen is in the A site, your stop codon will rest there. So for instance, in this particular case, we've got a UAA in this particular case. And instead of recruiting a tRNA with an amino acid, what it's going to recruit is a releasing factor, which is a protein, who will basically come in there and inhabit the A site. But you notice it doesn't have an amino acid attached to it. So here's your ribosome sitting around, already gone through codon recognition. We got that part. The releasing factor is sitting there, it's kind of squatting on the A site. And now it's like you're waiting for peptide transfer, right? That's the next step in elongation, peptide transfer. But the problem is there's no amino acid to transfer to. So you're kind of stuck. So as a ribosome, you sort of like pause a little bit. It's like you're jammed. And usually what happens is once you kind of get paused like that, any kind of an enzyme complex that has a pause like that, it'll kind of pause for a little bit, but then it'll basically abort mission. Because what it'll do, it'll be like, okay, obviously this isn't working, so we're just going to go ahead and fall apart. And that's essentially what always the response is once you kind of get those pregnant pauses in there. And so once it pauses, it's going to force the release of your polypeptide chain. It can go off into your 4 levels of protein folding at that point, and it's going to dissociate, right? So basically it means it's going to fall apart. Everything. So your tRNA, your small and large subunit, your mRNA, the whole kit and caboodle, it all falls apart. And then you basically get to go do it again and start over with initiation. And so this is what you do, right? You basically go and you translate, you fall apart, and then you reinitiate. Translate, fall apart, reinitiate. And you just do this over and over and over again. So one of the questions we have that was sort of hanging over, maybe not, but I had a question, right, is when we looked at chapter 4, right, we're looking at a secretory pathway. We looked at rough ER and the fact that you had ribosomes attached to the membrane of rough ER. There's a couple of questions there that are associated with that process, right? You basically kind of took it for granted that they're there, like the sort of the magic ribosome fairy just happened to put them there. And so that's obviously not the case, right? But there's 2 questions that come from that process. Number one is, what's the mechanism of how the ribosomes get themselves attached to the rough ER, right? Where we talked about the ribosomes, we talked about they could be attached to organelles. We also talked about they could be free, but we didn't really explain why or how they could be attached to the ER. This is one of the questions we get to answer now. The other question we actually have, though, in addition to that, is remember the rough ER is really targeted on very 3 main specific proteins. If you're associated with the ER, you're basically making one of 3 proteins. You're either making a secretory protein that be secreted to the outside cell, right? Or you're making a membrane protein. We talked a lot about that in chapter 5. Or you're going to be making a lysosomal protein. That's it. So the question is this: not only how does the ribosome tether to the ribosome or to the rough ER, but how does it know that it is choosing an mRNA that is one of those 3? How do you know to specifically get those 3 proteins associated with the rough ER? Okay, so what's your mechanism for deciphering which mRNA you have? Well, we answer the question here with protein targeting. It all basically wraps around our SRP, our signal recognition particle, which is a part of the mRNA. And it's a piece of sequence. We talked about this earlier in the chapter, and we promised that we'd come back to it, and here we are. But it becomes an actual signal on the polypeptide chain as you're producing it. Why? Because it's already there in the mRNA. So you're basically, as you translate it, you translate this particle that's attached to your polypeptide, and this acts like a zip code. Just like we use zip codes to localize ourselves, right? I mean, zip codes are an invention of the post office so that they can basically localize where you are, right? So at least they can get your mail somewhere near you, like in Denver, somewhere in Denver, as opposed to somewhere in Detroit, right? So they use zip codes to do that. But you use the same thing here. The way that you're able to localize these things is by using the signal, which acts like a little zip code. And then the other thing is, once you have this little signal that acts like a zip code, right, this signal recognition particle, you also need a receiver for that, right? That's where your ER receptor comes into play. So there's a receptor system on the ER that is receiving this signal peptide. That's always true, right? Wherever you have a signal, you have to have a receiver for that. It's a ligand receptor relationship. All right. And so the interaction between this receptor and the signal recognition particle is essentially how you dock the ribosome to the rough ER and essentially kickstart the secretory pathway, essentially. So that's how it works. So here's a ribosome happily going about its business, going through its translational business. It already did initiation. Now it's going through its 3 steps of elongation, right? And its little codon recognition, and then peptide transfer and translocation thing. It's doing that over and over again. One of the first things actually that was associated with this mRNA as it was translating is a little piece of signal that's on the actual polypeptide itself. That signal is your zip code. That is your localizer. That's what you're going to be scanning in order to tell you where you need to be going. Because what's going to recognize that is your signal recognition particle. Your signal recognition particle will come in. It will have an area on it that's designed to actually bind to and recognize that little signal on your polypeptide. When it does that, it will bind to your polypeptide and it will insert itself into the ribosome. So now what you have is a ribosome with like this little adapter attached to it. So what happens from there? Well, now we have to go and take a look at what the rough ER has. The rough ER has a protein channel, which you can see here, but it's attached to a receptor for the SRP. So the receptor for the SRP is associated with the protein channel. And what happens is this SRP will basically move and lock onto this receptor, kind of like the yin and yang symbols, right? Where the 2 of them come together to kind of form the whole. And when these 2 lock on, the SRP will then be embedded into the membrane, thereby anchoring it to the rough ER. And then through the protein channel, you'll start to thread your polypeptide chain as it's being made. So then what's happening is your polypeptide chain will enter into the lumen of the rough ER. And then once it's done, it'll basically start going through its 4 levels of folding inside the lumen of the rough ER. And there we go, away we go. Now we're going to go back to chapter 4 and start all over again with the secretory pathway, right? So that's where we take over. So this actually answers 2, well, at least 1 question, but not the other. It answers a question of how do you actually get the ribosome to attach to the rough ER? It's a simple signal receptor relationship, but it doesn't answer the question of how do you know which mRNA to choose? How do you know that this isn't a protein that this particular thing is making that's supposed to be free in the cytoplasm, right? So attaching it to the rough ER would be inappropriate. Well, it comes back to your signal, your little peptide signal. This is like a barcode. So in your mRNA, remember your mRNA has a couple of features to it when you take a look at it, right? We have our 5' cap in our mRNA. And we have our poly A tail. We have our open reading frame. That's going to be from protein. But typically in an mRNA, there's 2 other regions in there that are not ever becoming protein. That is to say, they're untranslated regions, right? So here you have like your start codon, maybe your stop codon here. So this region here is what's called the 5' untranslated region. There's also a 3' untranslated region. What you find in here is regulatory sequence. Because as you're manipulating and working with your mRNA, you have to have a place to be able to contact it. And that's what this 5' UTR is. So when you're basically regulating how your mRNA behaves after you make it, you use your 5' UTR to do that. A good example of that is where would you put different types of barcodes and things like that? Well, one potential place that you can do that is in your 5' UTR. Okay, you could have like a sticker that says, you know, airmail to nucleus, right? And then that's kind of like your zip code, and then other proteins can see that and be like, oh, okay, so you're going to go to the nucleus. So we'll make sure we get you to the nucleus. Another alternative is in this signal. The signal itself, like a zip code, will tell you where you need to go. So when the SRP comes along, there's other recognition particles that would say, like, listen, I'm not going to look for the sequence that's associated with the rough ER because this particular mRNA is going to be docked on the nucleus. So there's a different recognition particle that come in and recognize that, and it would do the same thing, but it would basically recognize nuclear receptors, right? So you have these signals embedded in your mRNA that's designed to be able to localize them where they need to go. Yeah. No, it stays there. Yeah, it'll stay there as long as it needs to until it's done, and then it'll probably fall off, right? Because it needs to go and reassemble. But the same thing's also true, right? So if you don't have anything to do, then you're kind of like, okay, abort mission, and you just fall apart, and then you just go reinitiate, and you start it all over again. Yep. So you can see, right? I mean, because back in the day, we used to think, and this is always how we used to think, right? We used to think that, it must be just kind of like random drift, right? The ribosomes must just sort of randomly drift and find their way to the rough ER. But we know enough about biology now to know that nothing is random, right? And usually when we start off that way, saying that this is random or just drifting randomly, we're almost always wrong. Because as we dig in a little bit deeper and we learn a little bit more, we realize, no, it's not actually random. It's actually a very, very intentional, very well-controlled and regulated pathway that we're seeing. Usually when we say it's random, it's because we don't know enough about the system. So then when we take a look at the chapter, we start off with transcription. So you can see here, you're basically making your RNA in your transcription bubble. A couple of pieces of nomenclature that I just want to sort of point out really quickly here is notice you have your template. That typically has a name we call the template strand. The alternative strand, oftentimes nomenclature-wise, we tend to call the coding strand. This oftentimes will confuse students. And the reason why they call it the coding strand is because this strand is essentially the same as your transcript. Think about it. Your transcript is a complement to your template, right? But so is the other strand. The other piece of DNA is also complementary to your template. The only difference is it's used for Ts. So essentially the reason why we call it the coding strand is because it looks exactly like your transcript, but you just swap out T's for U's. Okay. So that's your transcription. Once you get transcription, then you're going to go into modification, right? So you're going to cap it, tail it, splice it. At this particular point, you're going to basically create your open reading frame so you can get your nice translatable product. Then you're going to go through the events of initiation for translation. So you're going to export out your final mRNA after it's been spliced out into the cytoplasm, where you're going to get it tethered to a ribosome. So that's your initiation event, right? And then from initiation, you're moving into elongation. Your 3 steps of elongation. So you have your codon recognition, where you're recognizing the next codon in line, and then that's followed by peptide transfer. And then from peptide transfer, it's going to be moving into translocation, right? So that's basically your elongation pathway. And not only that, but this taken together essentially explains to us the idea of gene expression, right? So how do genes express themselves? They express themselves through the proteins that they encode. And a lot of times when we're trying to understand gene expression, we oftentimes think of it a little too simply. We think of it in terms of like, okay, so we turn the gene on, turn the gene off, make the protein, don't make the protein. But there's a lot more going on there. Now think about it. One of the things I want to challenge you to think about is think about all these processes we just went through for the majority of this chapter. Does it seem like a lot? There's a lot of steps in there. Does it seem unnecessarily busy? It seems like there should be a more efficient way, right? How about 1, maybe 2 steps? Boom, right? Cut to the chase, get to the bottom line. Wouldn't that be a good idea?
I feel like we miss a lot of spots.
And that's so each step, and this is always, you always want to think about cellular economy, right? Every time you do something, it costs you. You want to transcribe your RNA? Guess what? You have to make an RNA polymerase to do that. That's going to cost you, right? You want to do modification. You're going to have to cap it. That's going to cost you. You're going to have to tail it. That's going to cost you, right? Because you have to come up with enzymes and create enzymes that's going to be costly to create in order to do these things. You don't just get capping for free, right? So every single step, you're adding more to it. in order to accomplish that step. Then we get to splicing. And that's super costly, right? Because you got an entire spliceosome involved there. So that's really going to cost you. And we haven't even gotten to translation yet. So every single step that you have is costly. Now think about it for a second. Remember, the cell has no discretionary income and no discretionary time. What that means is, if it's something that costs you, then there's you look around, there is a huge upside that offsets that cost. It's always about cost benefit. And the one thing always remember, and the reason why I say always remember, the cell does not have discretionary income or discretionary time, is that whenever you see big, huge, multi-step pathways like this, and every single step is going to cost you because you have to come up with the labor and the mechanism and the chemistry in order to pull that task off before you even get to the next one. That's incredibly costly. So since you're spending so much money that you don't have, there'd better be a titanic upside. And there is. Because with gene expression comes gene regulation. Oh, wow, that was anticlimactic. That's the upside. That's what we're building up for. Yeah, basically, yeah. Okay, maybe that was, I have to go back to, yeah, that was, okay, let's go home now. That was it. That's all I got. No, anyway, right? So you have points of regulation here, right? So for instance, if you're regulating your gene expression at this level, this is what's called transcriptional control. We talked about that a lot, didn't we? This is that regulatory region that's just upstream of the promoter. In that regulatory region, you have all sorts of different little signals in there that will either dictate or determine how that gene behaves. And it's more than just lights on, lights off. Almost nothing is that simple when it comes to gene regulation. What you actually have is an entire battery of things that are more like dimmer switches. And you have multiple dimmer switches in there. So it's almost never, do I turn the gene on or off? It's usually, how much do I turn the gene off? Like, do I just give it just a little spritz, right? But it's not just that, it's also timing. Not just how much do I turn it on or off, but when. The timing is just as important, just like music, right? You have to know how loud you need to be when you chime in, but you also have to chime in on beat, right? Because otherwise, like, I mean, I kind of have an oboe at home that I play around with. And if you don't do that one right, it sounds like a dying duck. An oboe, it's kind of like a clarinet, only you don't ever call it a clarinet because an oboist will actually get angry at you if you call it. It's a double reed. Yeah, it's part of the woodwinds family of instruments. But it sounds like a duck. It sounds like a snake charmer, right? That's basically what it sounds like. But it's really loud. It can be quite garish if you don't have it just right. So you have to come in with just the right strength. So you have to soften it a bit, and you have to do it right on cue. Otherwise, everything's going to screw up. That's attenuation. That's what it's like for proteins. You have to come in right on time and just the right amount. And you have all these controllers in the regulatory region to control that. So that'd be transcriptional control. But what about if you control here? This is what's called post transcriptional control. So an example of this would be alternative splicing. That's a huge one, right? You got one gene, one transcript, you made it, and now you've got multiple isoforms because you have different splicing patterns that's going to generate different outcomes and different products for you, right? So that would be an example of post-transcriptional control. But then what about over here? Is there a way that we can affect translation itself? Sure there is. We do have translational control as well. Because just because you make the mRNA doesn't obligate you to translate it. You can translate a little bit of it, or all of it, or you can store it. It's all up to you. If you do that, then that's translational controlled expression. So for instance, one strategy that you can do is you can make a ton of mRNA, have it all ready, but you don't want to make it just yet because you want it to be ready, but you're going to maybe store it or protect it, like maybe fold it up in hairpins to kind of protect it from degradation and things like that. But when the environmental cues are just right and the circumstances are just right, you want those ribosomes to start going full tilt and making all of your translation product from these mRNAs. But you have to wait until the environment gives you that cue, right? That's an example of translational control. Affecting the overall expression of that gene. Is that it? No, of course not. That's never it, right? You also have post-translational control. An example would be glycoproteins. Post-translational modifications, right? Basically, a glycoprotein is a protein, goes to the Golgi modifies it by putting a big old carbohydrate branch on it, and then it's functional, isn't it? So that carbohydrate addition is a post-translational modification. And you can get an entire group of different types of behaviors just by monkeying with those carbohydrates, right? We saw that, didn't we? In our ABO blood group. All we did there was alter the different types of carbohydrates that were sticking out there. That's all we did. And it had titanic differences in the expression of the ABO genes. Okay, so that would be post-translational. So there it is, all this translational control, this post-transcriptional, transcriptional, translational, post-translational control. All that added together basically affects gene expression, how a gene ultimately is seen in your overall phenotype at the end of the day. But wait a minute, wait. I thought you said this was an upside. I mean, what I'm seeing here is more cost. Because in order to have these controls, you have to have proteins that do the controlling, which means you just spend a lot more money on all these transcriptional regulators and post-transcriptional regulators. You spend a lot of money on the spliceosome. You spend a lot of money on these translational controllers. So I don't see benefits. I see racking up huge costs. If anything, you're digging the hole deeper, right? So I ask you again, what's the upside? If you're laying out all this cost and you don't have a lot of cost to lay out, then there has got to be a titanic upside. What is it? Yeah, which we can bundle in a couple of different synonyms, right? Variation, diversity, but variation. Notice we've been down this road before, haven't we? We figured out that the reason why we're willing to put up with sexual reproduction, even though it's not the most efficient or the most energetically cost-effective way to do things, what we got for it was infinitely beneficial, right? So that was from a genetic sense, but this is from an expression sense. So it is genetic, certainly, but now you have another layer of variation on top of the genetics. So not only do you have a gene firing off a protein, but now you have all these regulatory controls that affects and attenuates actually how that gene is active. So basically you have variation on top of variation. What does that do for you? It gives you, when you combine it all together, it gives you an infinite possibilities to be able to do what? To adapt, right? Because what's your environment always doing? That's all it does, right? So if you're not ready for the environment to throw you a right hook, you're waiting for the jab, guess what? You'd better have a strategy for that. The more variation you have, the more expressional variation you have, the more likely you're going to have some sort of a response for that. So rather than standing flat-footed with a glass jaw, taking a right hook square to the mug, that's going to put you on the floor. Guess what? That's a failed mission, right? What's the purpose of biology? Okay, that's nice, but not the class biology, but like a biological organism. To reproduce, right? Everything you do is for that purpose. Guess what? You got to live long enough to do that, don't you? The only way you're going to live long enough is if you're ready for that right hook. That means you need variation upon variation. You've got genetic variation, but that's not going to help you, is it? Your genetic variation is going to help your descendants, because it's your meiosis that's scrambling the genetics that benefits your progeny. That's not going to help you, is it? What will help you is your gene expression. So the genetics that you do have, that you're stuck with, can be expressed with a lot of variation so that you have a lot of tricks in your bag to be able to adapt to the environment. The more tricks you have, the longer you live, the more you reproduce, and you basically dominate the world, like Genghis Khan, who apparently is pretty much everybody in Asia is related to the guy. Apparently he was a randy little mutt, apparently, as the stories go. Yeah, let's compare them, right? So prokaryotes versus eukaryotes. Prokaryotes don't have any introns, eukaryotes do, so it's not a prokaryotic thing. How many genes do you see in an mRNA, right? This gets back to the operon structure of the prokaryotes. Remember that polycistronic mRNA where you're rattling off A, B, and C. We have one gene, one mRNA, right? So it's kind of a different structure that way. In terms of transcription, our transcription is occurring in the nucleus. Our translation is happening in the cytoplasm, right? So it's all happening in the same place in prokaryotes. We both have the same start codon, but the difference is we have a 5' cap with a normal methionine, whereas bacteria have that informal methionine. So their methionine is slightly modified. It's a little bit different. And of course, in terms of modification, bacteria, prokaryotes don't have any modification, right? They don't have any RNA splicing and stuff like that like we do. Okay. So what happens if we screw it up? Mutations, right? So this is basically an alteration of your DNA sequence. And so what you take a look at is basically what happens when you alter the actual DNA sequence. Like for instance, if we change a single base pair, right? So if it's supposed to be an AT pairing, what if we change it to a CG pairing? What's the consequences there? So what we have here is a list, first of all, of increasingly more severe mutations. In this case are what's called point mutations. So these are all mutations that occur just by changing one single base pair. And that's it. The first type of point mutation is called a silent mutation. Now, this is basically one where you have the same amino acid. So there's no change. And we see this, right? Because if you take a look at the code table, you'll notice that some of these amino acids actually have several codons that encode for them, right? And so they basically have, like the lysine, for instance, I think has 4 different codons. So if you were to mutate from one of those codons to the other codon, you might get a change in your DNA, but you don't get a change in your protein. So it's functionally, from a protein perspective, it's actually, sorry, my OCD coming in. It's actually no change in the protein. So you have no visible change in the protein. That's why it's called silent, okay? Now, what happens if it does change the amino acid? This is going to change your amino acid designation from one amino acid to another. Well, we have 2 different types. We have what's called transitions, which is essentially when you change a purine to a purine. So like that's an A to G type of situation, or a pyrimidine to a pyrimidine, right? That would be like a T to C type of a change, okay? So those typically are a little less severe, but you will change the amino acid, and that could be a problem, right? So it depends on where that change happens. Mutations can, even though if there are multiple mutations in a single gene, a mutation is not the same as other mutations. So it depends on where it is and whether or not that part of the gene is tolerant to that change, okay? Transversions are a little more aggressive. This is basically when you're changing from a purine to a pyrimidine. And this causes more structural disturbance in the actual DNA molecule, because you're going from the double dog dish of a purine to the single dog dish of a pyrimidine. So that's enough to create a little bit of a structural change in just the 3-dimensionality of how those fit, okay? Then you're going to have the nonsense mutations. This is a situation where you make a base pair change and it actually turns it into a stop codon. So like, for instance, if I have a UCA and I change that C to a G, then all of a sudden UGA becomes a stop codon, right? So what that does is basically tends to truncate the protein. And you pretty much get very little to no enzyme activity, if that's the case. Okay. So here's kind of what it looks like. So here's our normal protein. You can see we've got our coding strand, our template, and here's our mRNA. So we've got our AUG, CCU, which is a proline, UAU, which is the threonine, CGC, which is an arginine, of course, UGA, go Bulldogs. That is our stop codon, right? Now, what happens if we change the CG? So we're going to take a look at this one first. So if we do a CG change, in this particular case, we're still going to have our AUG, so that's our methionine, and our proline is still there. So CCC versus CCU, that's a codon that basically encodes for the same amino acid, proline. You got lucky here. You changed your codon, but you didn't change your amino acid. So that's a silent mutation. So no change in your protein. As a matter of fact, this also represents one particular type of polymorphism between people, right? So we accumulate silent mutations randomly. The patterns that we accumulate are all different from each other. So if we were to analyze my silent mutations, they would look different than yours. And I inherit my silent mutations from my parents, and the same thing you do from yours as well. So we can actually look at these silent mutation variations as almost like genes and kind of trace our ancestry with them. Let's take a look at a missense. So in the case of a missense, we're going to take this and we're going to convert this one right here. So notice originally you have a methionine, proline, threonine, and it's supposed to be arginine. But what happens is instead of arginine, CAC encodes a histidine. So here you have a change in your amino acid. Now, whether or not that is tolerated totally depends on where in the protein it happens. If it's in an area of the protein where, you know, it's not a very big deal, then you may be able to get away with it, right? If this change happens in an area that's critical, like the catalytic domain, where you basically, this change sort of screws up critical interactions that is in charge of its catalytic activity, then this could be a catastrophic change. And it just depends on the nature of where it is. Now, the nonsense mutation, basically, if we change this one here, notice we're changing it from, instead of UAU, we're changing to UAA. Notice it encodes a stop codon, not codeine, a stop codon. Codeines for a different day. But anyway, you still get your methionine and your proline, but now notice you get nothing else. So this is your truncated protein. And this is usually pretty deleterious. You don't really get a whole lot of rescue from this one. Okay, so this one's pretty well dead. Yeah. The truncated one? This one? Yeah. Because all you have is just 2 amino acids. So it's like literally like the protein doesn't even get made. So, and that's usually pretty catastrophic, because that means you're not making the most important parts of the protein. Yes, methionine is always the start codon. Every single protein starts with methionine because that's our start codon, AUG. Getting more aggressive are frame shifts. These are really nasty. And we already talked about them, right? So I don't want to kind of belabor that because we talked about them when we were talking about your genetic code being read as 3-letter words, right? So we kind of figured that 3-letter word piece, we're playing around frame shift mutations. So anytime you're either adding or deleting a single or 2 base pairs, you're going to get a frame shift. This will create a lot of, like, when it shifts the frame, it just creates these sort of nonsensical amino acids. It's just nothing, right? It's just gobbledygook. And oftentimes will create premature stops. Eventually what's going to happen is it'll create a premature stop. And so not only are they truncated themselves oftentimes, but what is there is just complete nonsense. Okay. There's also another type of mutation that we don't really know where to put it, but we put it under frame shifts. And that's a triplet repeat expansion. And basically in some genes, not all genes, there are triplet repeats. So like, for instance, a triplet would be like a CGC triplet, but it repeats. So you have a CGC and a CGC and a CGC and a CGC and so forth. And so you have these kind of repeated units in this particular gene or in these particular genes. A good example is in the Huntington gene that underscores the Huntington's disease. And the interesting thing about these is they're really, really curious. They don't behave normally because in some families, they expand. That is to say, you get more. We don't really know what causes that exactly, but we think it might have something to do with inaccuracies or inefficiencies in replication and how they deal with these long stretches of repeats. But from in any case, basically, they expand and usually expand from one generation to the next. So for instance, if I have a CGC repeat, and let's say I have 10 of them, then I have basically, I'm normal, right? So it's in the middle of a gene, and so it's not causing a whole lot of problems sitting there in the middle of that gene. However, the next generation will see an expansion into 20. And so they'll basically move into a borderline scenario, not necessarily full blown, but they're kind of borderline. And then their kids, the grandkids, will expand even more. And then they'll get the full blown disease. And so that tends to expand over time. And that expansion creates problems because as you're expanding these repeats, what it's doing is it's in the middle of a gene, it just basically blows the gene up, right? So it can't function normally anymore. And so there's a lot of research going on in these triplet repeats, and we're trying to understand how they work and behave because they are quite different in the way they behave than anything we've seen before. Now, in the genetics class, we actually have two different chapters in genetics class we cover on mutations, right? One on micromutations, like alterations in sequence and how we deal with it. And the other one is on chromosomal mutations, so abnormalities on a chromosomal level. So you can do the same things on a chromosomal level, only in this case, instead of doing a deletion of a piece of sequence, you're doing an entire chunk of a chromosome is gone. Or a duplication, instead of duplicating a little piece of sequence, you're actually duplicating an entire chunk of the chromosome, okay? An inversion is an interesting one. We haven't really talked about that one, but basically what an inversion is a region of a chromosome that gets cut out and it gets spun around 180 degrees. So like if everything's being read in this direction, when it gets spun around 180 degrees, it basically gets reinserted backwards, inverted from where it normally would be. And so that's your inversion. Of course, translocation is basically where two chromosomes swap pieces between each other. And so we'll take a look at each of these here. So here's a deletion. So for instance, when you take a look at a deletion, we have a chromosome here, A, B, C, D, E, F, G, H, I, J. So here you have your centromere. This is your long arm, typically referred to as the Q arm, and then this is P, usually the short arm. That's kind of a human genetics notational sort of a thing. But when we delete it, we're going to be working on the BCD region. So we're going to basically take this region and delete it out. And then what you get is AEF. So you had BCD, used to be right in that area. Now, the important thing is to remember is that when we're talking about BCD, we're not talking about genes. We're talking about regions of a chromosome with hundreds or thousands of genes. So you're not just talking about individual genes, you're talking about entire regions of chromosomes that you're missing. So chromosomal level mutations are pretty catastrophic, right? Because you're losing a ton of information in this case. Or if you're duplication, right? If you duplicate your BCD, you can see you got basically a BCD, just a little bit of a stutter, you have a BCD and EFG. So in this case, you have too much. So you have double copies of those hundreds to thousands of genes. And remember, in animals in particular, We don't tolerate extra DNA, right? So where we talked about non-disjunction and how losing a chromosome is catastrophic and gaining an extra chromosome is catastrophic. So we don't tolerate changes in the amount of anything. And so that's the reason why these are pretty bad.
Is that how 1920 works? You're talking about it adds on more.
So that's the repeat is a little bit different because that's more of an expansion. So it's kind of like, generally speaking, with that one, it's more like the replication machinery sort of is doing these CGCs, and you can imagine if you were reading through it, you would lose your place pretty easily, right? Because like, wait a minute, was this the fourth or the fifth CGC, right? So, and if you miss that, then you could step back and then duplicate it again. So like you could do 1, 2, 3, 4, 4 again, 5 again, right? You're kind of constantly losing your place because everything looks the same in these big repeat islands. And that's what we think is happening. Well, the chromosome is big, so it's going to be hard to actually see a longer chromosome from just triplet expansion. It would have to expand quite a bit in order to see a visible extension of the chromosome. So what about an inversion? So an inversion in this particular case, we're going to be cutting out B, C, and D, spin it around and put it back in, right? So here you can see you got A, and then you can see B, C, and D is backward now. And you have E, F, G, H, I, J. But guess what? This isn't actually accurate. To be a real inversion, what you'd have to do is you'd have to basically write these letters backwards. Because that's actually inverted. That's going the opposite direction. But of course, you can't do that with graphics because there's not a typeset function to say, just write these backwards. Can't do that, right? So it actually looked like this. Now notice that the genes that are in there are normal. They're there. You're not missing anything. They're not duplicated. They're just pointing the other direction. For the most part, there's no harm, no foul there, right? So it doesn't really matter which direction you're pointed. You'll still function normally. What makes an inversion bad, and a translocation, by the way, is not necessarily the genes being inverted as much as where the break points occur. The break points, if they're in bad areas, are basically taking pieces of DNA that live in one neighborhood and moving them into another neighborhood where it may not work, right? So for instance, if you have a regulatory region for a gene that's always on, and if you cut it right there and flip them around and then all of a sudden put them in front of a gene that's normally off, then all of a sudden that gene that's normally supposed to be off gets turned on because it's got abnormal regulation behind it now because of the breakpoints. So what you see is dysregulation of the genes in and around the breakpoints, and that causes the problems with both inversions and also with translocation. And a translocation, it's pretty easy. Basically, you have A, B, C, D, E, F, G, H, I, J, and you have KLM and O, P, Q, R. And what they're going to do is they're just going to break off a chunk and swap it. So ABC and KLM are going to be the ones. KLM is going to basically grab onto the ABC chromosome, and then the ABC is going to grab onto the KLM chromosome. And so here you can see again, the genes in there are normal. They don't really care where they are. They're there, they're functional. And so you don't have extras and you're not missing anything. But again, where it comes down to, for both of these, is the problems are with the breakpoints. And actually a good example of the translocation, the way we actually discovered translocations in the 80s was in studying leukemia. Leukemia, being a cancer, is supposed to be random and stochastic, right? So you're unlucky, you happen to get a bad combination of mutations and you got leukemia. That's usually how it works. What we noticed, however, was in a family in Philadelphia, they seemed to be exhibiting an inheritable form of leukemia, which was puzzling to us because it's not supposed to be heritable. So we tried to figure this out, and what we realized is that what was causing it was a translocation. Though what happened was a breakpoint, which picked up a regulatory region, got plopped in front of a gene that caused leukemia. And we called that chromosome the Philadelphia chromosome. And that created familial leukemia. And that's how we figured out translocations, that they can actually be quite noxious in nature and cause lots of problems because those breakpoints are very, very particular about where they land. And so there's obviously, if you that's a fascinating story. They have an entire chapter on that in the genetics class. Now mutations themselves, because that's what this has been about, right? So we have micro mutations and sequence mutations, abnormalities in chromosomes. And the importance of mutations is that the idea is that this is kind of like your starting point for evolution, right? This is basically your source of new information. And actually mutation is one of the mechanisms of evolution. There's several. This is one of them. However, we all know exactly how I feel about mutation, right? I mean, it is a thing, but here's the problem. They're overwhelmingly negative, which is a problem, right? Because the idea is if all of our genetic variation came by way of mutation, then how do we answer this question, right? Because it seems to be counter to how mutations actually act. Because if you think about it, like a mutation, for the most part, blows stuff up. That's what it does, right? I mean, we have an entire human genetics catalog of genetic mutations. I challenge anybody to tell me of a mutation that was a good thing. Teenage Mutant Ninja Turtles. Okay, so that's a cartoon. So something that's actually real. Because think about it, right? Mutations are blowing stuff up, right? So that's what they do most of the time. But go ahead, Raphael. So that's kind of, so there's an interpretational overlay to this as well, right? Because the idea is if you're trying to figure out beneficial mutations, because that's what the idea was, right? The idea isn't, oh, we don't acknowledge or not acknowledge the fact that mutations are largely negative. We acknowledge that, right? The idea is that the beneficial ones accumulate over vast expanses of time in order to create our collection that we have now. The problem, and that's fine, but that's hypothetical, right? Because we haven't actually discovered a beneficial mutation in human history, right? So the ones that most evolutionary biologists use as beneficial are largely interpretational. It's like, it's beneficial because you sort of, kind of, right? So you sort of overlaid a benefit to this and it turned out to be a good thing, right? So that's not the same thing as being truly wild type beneficial, because overwhelmingly it's negative. And so the rate of beneficial mutation is incredibly slow.
What about that dude that injects himself with snake lines every day for like a year so that he provides himself with the snake and then time? That doesn't have like mutate your reason to make me
No, that's just developing your immune system.
Yeah.
That's a totally different mechanism.
Every day for a year, a little bit till you up the dose to full vitamins, the different vitamins.
That's just training your immune system. That's no different than basically exposing yourself to chickenpox. That's basically what you're doing. You're just training your immune system to prepare itself and be ready for that. So your immune system will attack anything. Like a tolerance. Yeah, similar kind of an idea, right? So it's not necessarily an immune system thing there, but it's a very similar type of a training, sort of physiological tonality to it, right? You're just basically training your body to accept a new norm, right? That's usually what's happening when you do subacute levels and you do it for a long period of time. All you're doing is retraining your physiology to recalibrate what normal is. That's what that is. No, because if they did, he'd be a train wreck. Because mutations are mostly deleterious, right? To the point where it's like, we could say like, okay, how many of you guys want to sign up for a mutation? Nobody's like, pass. Why? Because they're pretty much dominantly negative. Nothing good comes from them. And that's the paradox, right? That's the paradox that I kind of always struggle with. Is like, yeah, mutation is a thing. I'm not necessarily saying it's so, but we got some work to do on that one because we still have yet to actually make that case. I feel like that case has still to be made, that this has the capacity and the robustness to actually accumulate a lot of beneficial mutations like this. Over how much time, I don't know, because we're still a little slushy on our mutation rates. Like how many beneficial mutations you get every million years or whatever, right? I mean, there's numbers all over the place. So I kind of feel like mutations are an important piece of the puzzle, but it's one that we don't understand as well as we want people to believe we understand. And that's the part that gets me. I don't mind the part of not understanding it or whatever. What I do mind, however, is for a lot of evolutionary biologists to basically put it out there like we actually do know something we don't, because I see that as a lie. That's what I see. But yeah, well, no, kind of going backwards. So that would be kind of different. So CRISPR is also an engineered sort of thing. So we're not creating mutation. We're actually creating the normal. We're replacing the mutation with the normal. So it's different. So it's a little bit different that way. None of that, but we're talking about mutation. I'm talking about natural, like development, not stuff that we can create in the lab, because we can mess with this all day. But stuff that just naturally occurred. We're talking about like out in the wild, not even our own dogs and things like that, but stuff that's out there in the wild. Like, what does that look like? And I don't know that anybody really has a clear idea what that looks like.
Much taller and much shorter.
Well, I mean, it depends on how you define mutation. I mean, if you define mutation as an aberration from norm, whatever that is, then yeah, right. But usually we define mutation within the context of survivability and natural selection. And if that's the case, which is how most evolutionary biologists explain it, then I'd say pretty darn close to almost 100% of the mutations, like 99.9% of them, are clearly negative, because almost none of them give you an adaptive advantage. Like it's something that compromises your ability, right? So we just talked about a whole catalog of them. None of them are good, right?
So
You're saying
that's overwhelmingly negative towards humans.
not just humans, any organism. Just mutation in general is generally catastrophic, because it's kind of like basically throwing, a nuclear bomb at a city like LA, like totally just torching it and then hoping in the aftermath it just sort of comes together as something pristine and beautiful and perfect, right? So it's kind of not going to happen that way, because mutations are destructive and they leave chaos in their wake. I mean, is it possible? Yeah, I guess it's possible, but I mean, are you willing to put money on the table for that one?
Wasn't borders like current?
No. Mm-mm. And this is, I want you to hang on to that one, because when we're talking about mutation, we're talking about new variation, right? So that's the new information. And historically, now what I'm pushing back on is Darwinian gradualists. Remember, I told you I'm a punctuated equilibrium guy. So mutation historically has been thought of as the source of new variation, new information, right? So for instance, as opposed to, I'm going to explain what I mean here. Old variation, which comes from pre-existing variation. Now, here's what I mean by pre-existing. When you see most evolutionary studies, most of them aren't starting off 4 billion years ago at the beginning, right? Most evolutionary studies are usually starting off somewhere in the middle. Like they'll start off at like the family level, or like the order or the class level. That's kind of where they'll start, and they'll kind of look at speciation from that point forward. And when you do that, what you're doing is drawing a line. And and in that line, you have a handful of genetic variation that's already there. That's what I mean by pre-existing variation. So if you're starting an evolutionary study from the point of family, then you're starting with a collection of genetic information already at that family level, right? And moving forward with speciation. And so here's how phylogeny works. Basically, you start off at the family level. And eventually what's going to happen is you're going to have a branch come off. And that'll be caused by a mutation, right? This is kind of a Darwinian gradualist sort of point of view. And so then you'll have another one kind of come off there, and that'll be caused by another mutation. Another one come off would be caused by another mutation. Maybe these two branch off, there's a mutation there. Another one comes off like that, and there's a mutation there. So now what you have is all of these species now. Here's 1, 2, 3, 4, 5, and 6. But when you do these phylogeny studies, notice you don't have any of this. You might have some fossils, maybe. But I mean, fossils are a little dodgy. You know how I feel about fossils, the rock zoo, right? But what you really have and know well is this stuff here. A bunch of animals that now live that have various morphological associations with each other. And then you kind of piece them together to sort of cluster them in terms of their morphological likeness. The idea being that with every new branch comes a new mutation. So from a Darwinian gradualist point of view, you had to, in order to get here, you had to accumulate 1, 2, 3 different mutations over time in order to generate these species. But here's the problem. If you already have this whole handful of genetic variation in the family level, because that's where we're starting from, going down, you already have sufficient variation in the family level to adaptively radiate to most current species. Now, that's a bit of a blanket statement. You have to actually study different branches, and different branches will show different things. But generally speaking, when you're talking about just a family level, there is plenty of information that's already there. When you just basically draw the line there and you just collect up all you have, there's enough genetic variation just in that to where you don't need a single mutation to get to today's species. Why? Because as you are adapting to your environments, that is chiseling away through natural selection, chiseling away and sort of creating different pools of genetics. Different generations will come up with different mixes of the same variation, and they'll start to slowly drift off and sort of differentiate from each other. A good example of that in human populations is Scandinavia, right? Where you have a preponderance of blonde hair, blue eyes, light skin, but that is nowhere near the dominant trait in human beings. But what happened was, in population genetics, is that group split off, and they had that collection of the genetic variation that then they perpetuated in those areas. And that's the reason why you see that. Same thing's also true. You mentioned bears, right? Bears is a good example, because the bears are all hybridizable. So you can take brown bears and you can mate them to polar bears, and you can make them to black bears. They're all hybridizable. It just happens to be, koalas are a little bit of an offshoot. I don't know that koalas are, but the big bears, the major bears. So those are considered separate species. But if you take a look at the actual bear family, there's sufficient genetic information there to basically adaptively radiate. So the variation that gave polar bears white fur was naturally selected for in polar regions.
Like the cats.
And the other good example is our dogs.
Yeah.
Right? We go from a dog, which was pretty generic if you take a look at the old Egyptian dogs, and then you turn it into a teacup poodle, which you can hold in your hand. And I know you can do this because my dad had a teacup poodle, and literally he stood in my hand. He was cute, but he was little, right? Or a mastiff. And they're all the same species. And if you consider all the variation within that, that all comes not from mutation, but from just natural variation that we selected for through artificial selection.
It's like 3 puzzles in one.
And you can adapt them to different circumstances. And that's exactly what we did, right? We've got hunting dogs that we bred for a particular hunt, right? The Mastiffs are bear hunters, so they're huge, right? And stocky. And the teacup poodles are, well, I don't know what they do really, but yeah, they're parlor dogs, right? So the queen likes them, and corgis. So that's where they come from, right? So that's their thing. We bred them for that purpose, but they're all the same species. But that gives you an idea of just how deep that natural variation is. As a matter of fact, actually, with dogs, to be honest, to be fair, there are some mutations in there, right? So it's a combination of both of these, actually. Probably it's a combination of mutation and adaptive radiation from existing variation. Because the smash face dogs, that's a mutation, right? And we think it's cute, so we bred it, and that becomes a thing, right? So that's an example of a mutation that became part of the pantheon. Okay. Now, would that be a benefit in the wild? Absolutely not, because those dogs have some tremendous problems with smelling and their nasal cavity. They have a lot of problems there. It would not play well in the environment, right? But that's a good example. Now, here's another pushback point. Let's take a look at the bears. The bears you can hybridize to each other, but they're different species, right? Polar bear is a different species from the grizzly, different species from the black. We do that on morphology. But if you want to use morphology as your rule, is there anything more morphologically different than a teacup poodle from a mastiff? So why isn't a teacup poodle a separate species from the mastiff? So you can see there's a problem with our definition of species and with our utility of species. And morphologically, you're different. And so a lot of times what we do is we have species and they're all different. And so basically we just say this is a different species, right?
Isn't it just like that?
Even though they might interbreed. And the other thing is also, a lot of students don't know this, but there's actually 6 different definitions for species in evolution. Which one you choose will change your outcomes.
Isn't it just honestly speaking like from the same family?
It's variation.
But we like ways.
Yeah. So it's variation. So like somebody who was like a strict variation person would say like there's actually not 3 species of bears. They're all the same species of bear. There's just variants, just like a teacup poodle and a bull mastiff, right? But you can see that and it's not an easy answer, right? The reason why I do this is to sort of show you a little bit about some of the complexities of evolution. Because when you talk to somebody, and this is what drives me crazy, right? A lot of times evolutionary biologists would be like, oh yeah, this was a mutation and it all came by new variation and here it is and blah, blah, blah. And it's like, well, wait. You're like super oversimplifying it because it's not as clear as all that. And there's still a lot of questions that's associated with that. Like, how does adaptive radiation and mutation work together, right? So, and how much of evolution from family down is adaptive radiation and not new mutation, right? So there's a lot of questions there. And honestly, we don't know things as well as we want everybody to believe we know. That's the thing that chaps me. This is the reason why I have such a grind on evolutionary biology, because evolution is fascinating. There's a lot of moving parts, a lot of big questions that we're trying to answer, but it drives me nuts because 9 times out of 10, so a lot of evolutionary biologists will basically say, oh yeah, we know the mutation's a mechanism and this is basically what no, it's not that easy. You don't know it as well as you do actually say you do. When you say you know that for certainty, you're actually lying, because when you get into the nuances of it and all these questions, there's a lot of going on here, and it's a little disingenuous to kind of tell people that we know this as well as we do when we actually don't, right? That's my point. Stop lying and be honest about your science. That's been my message the entire semester. If you don't know it, then say you don't know it. Don't draw a picture of a dinosaur, you know, that's warm-blooded and crapping somewhere or something like that in a big forest of cycads, because that's the way you think it was back in those days, right? That's artist impression, not a scientific fact. Museums drive me crazy. Yeah, well, they do, because what they're trying to do is educate the public, right? So they want to basically create a holistic story that will get them money, right? And people don't give money to stuff that you don't know. If you say like, I don't really know what it was like back then, nobody's gonna give you money for that. But if you paint this rosy picture, it's like, this is what it looked like, and here it is, and this is what, well, how do you know that? It's usually based on a lot of interpretation and artists fill in, and not a lot of scientific certainty. Because even when you get into the science of it, it's like, that could be one of the, it could have been what it looked like. But there's also a lot of other possibilities how it could have looked like, and you're not being honest about that. Right? That's my big problem. It's kind of like when I go to museums, I feel like they're lying to me, and they're intentionally trying to dupe me. And I know better. That pisses me off. Because, yeah, because the questions are actually more interesting than the propaganda. Because they're fascinating. But and I hate that, because I think a lot of people have a very sort of preschool view of evolution, honestly. And evolution is like one of those things that needs to be done, and done well and thoroughly. Okay, that's it. That's the end. Go home. No, seriously. Go home. No, that's it. Yeah.
Quit saying you're kidding.
Yeah, because we're not.